So welcome to our Docker webinar. So just a couple notes. We won't be, because there is a delay with the On24 platform, like basically you see things about 15 seconds after we talk. We won't be doing that much live interaction, like answering questions like while we go. We'll do our best to answer questions at the end of the session. We just want to kind of not have it be confusing that, like, because of the delay on your end. Okay. So my name is Ted Lederis, and my colleague is?
My name is Andrzej Vampir, and I'm Community Engagement Scientist at DNAnexus, mostly focused on UK-Europe data.
Great. So let's talk about the formal learning objectives for the session. So number one, explain the benefits of using Docker on the research analysis platform for reproducibility and for batch processing. Number two, define the terms image, container, and snapshot in the context of Docker. So I have kind of gone through, a lot of, the literature. Like, these terms are kind of inconsistently used. So we're defining them in this way for this webinar, just so that we're clear on the concepts. So number three, so we'll be able to create snapshots on the research analysis platform using Docker pull and Docker save with the TTYD app. We'll talk about that in just a bit. Once we actually have these containers available to us, we'll utilize them to batch process files on RAP. And then we'll show you how to extend a Docker image by installing software within interactive mode. And then we'll discuss further applications of Docker on the platform. So we will mostly be talking about doing executions using apps and Swiss Army knife, but there are other ways you can use Docker on the platform that are also helpful. So we just wanted to be clear that this is what we're not going to cover. So Docker, using Docker Compose on RAP and running Kubernetes clusters on RAP. So we have not kind of encountered like good use case examples to kind of do this. So that's why we're not covering this today. Also, you can imagine these two could be topics on their own. So let's just kind of talk about the motivation for Docker. So number one is the idea of portability. So with Docker, the software environment is such that you can run code and scripts on any machine that has Docker installed. And this is really important for kind of cloud-based work, is that you avoid the installation headaches on running things on multiple machines. So this kind of part of Docker, the portability, really enables us to do batch processing without having to worry about installing, for example, SAM tools or BCF tools all on the multiple nodes. So this really, on my side, kind of the point of this is really to make it easy to run batch jobs on multiple instances. So let's talk about the reproducibility side. So, this is near and dear to my heart. I've taught courses in reproducibility. So basically, the idea of reproducibility is being able to run code and generate the same outputs given a set of input files. And that's part of the way we do that is tying all of our software to specific versions. And once you have those kind of versions locked down, you can utilize the Docker images. So we're going to be working with a tool called Swiss Army Knife on the platform, but Andre is also going to show you how to install another sequence analysis tool called EMBOSS. So some examples of reproducibility, definitely if you're an R person, the Rocker project. Of course, the genome analysis toolkit is also Dockerized. So these are all very good kind of resources in terms of Docker. So I think part of the hardest thing about learning Docker is all of the terminology. I'm going through working on the Amazon certification exam, and I think part of it, the hardest thing about it is knowing all of the terms. So let's start with the idea of a registry. So a registry itself is a collection of repositories that hold container images. And we'll be getting into the definitions of containers and images in just a second. But these are things such as key.io, QUAY.io, or Docker Hub. And the way we interact with these, so usually what we're going to be doing is we're going to be doing a Docker pull of an image off of one of these registries onto our computer. Or in our case, we're going to basically be using a web-enabled computer when we do this. And when we so we might make some changes. So we might, when we make those changes to the installation, we can also commit those to the image in Docker Hub. So these are kind of our two main tools for interacting with a registry. Okay, so this didn't quite turn out. So let's talk about terminology. And again, like I have kind of looked at multiple places, and we use this kind of in a little bit of an inconsistent way. So I'm going to call the Docker image. So that's going to be the, quote, recipe for building the software environment. So this is what we're going to use Docker pull to get it out of the registry. To actually generate these images, we're going to be using a Dockerfile. We'll talk more about that a little later, but let's just kind of focus on images and kind of what their instantiation is. So to pull an image, we're going to do this, as the code says here, we'll do a Docker pull, and then we need to give it the name of the, basically the name of the registry and the owner of the image. Once we have that pulled onto our machine, we'll call that a Docker container. And so that's the actual executable software environment that's installed on your machine. So again, we get the Docker container from a Docker image using Docker pull. And then once if we make any changes, we can basically, so we want to basically save this as like the software environment as a single file. So we're going to call that a snapshot file. So when we talk about snapshot files, we're going to mention they're going to be single archives. So like a .tar.gz file that contains the Docker container. So our main mechanism for generating this is going to be using Docker save. So what we have some kind of advice, and most of our advice centers around using snapshot files rather than using pulling the image every time, and there's some reasons for that. So, like I mentioned, we give you the advice of really using these snapshot files instead of putting in like the image URL here. Because there's a pull limit on Docker Hub and other registries of 200 pulls per user per day. So if you're doing batch processing, this is a big limitation. So basically, you're going to be limited when you basically use the URL. So we're going to show you kind of how to work with saving a snapshot file in your project. So this lets you kind of overcome these limits. And especially, like we've been mentioning with batch processing, it's especially helpful. So we would be remiss if we didn't talk a little bit about security. So this is something that you should be very aware of, and we try to give some advice about this. So when in general, if you can use images from trusted vendors whenever possible, like these trusted vendors have done a lot of the security vulnerability scanning and basically kind of avoid a lot of the kind of possible vulnerabilities. So they have a security team attached to it. So being aware of using trusted vendor images. So the other side of that is to kind of avoid using kitchen sink images. I mean, the temptation is to kind of use, some like a bunch of images that are built on a bunch of different kind of Docker images But I will say that, part of this is that it's hard to kind of manage all of the vulnerabilities if you're kind of installing things from multiple places. So one way to be very certain is that you want to scan your images for vulnerabilities. This is a link to a container scanner called SNCC, and basically you point it to the image in your Docker Hub or your key.io registry, and it will let you know of any vulnerabilities. So, I just wanted to give an example of a vulnerability that we all are probably familiar with, which affected quite a few different kind of installations, which was log4j. So there was a vulnerability in log4j, and that affected quite a few different web stacks and things like that. So just be very careful when you're kind of building your image files. If necessary, use Dockerfiles to uninstall and patch possible vulnerabilities in your image. We'll be talking about Dockerfiles in a little bit. Oops. So just a quick summary of what we've learned so far. So we talked about Docker containers and the ability to reproduce work. We can pull Docker images from a registry to basically make a container on a machine. And then we can make changes to a container. We can save it as a snapshot file. Andre will be showing you that in a little bit. And working with the snapshot files is really best practice on the platform. And of course, being aware of your images and when possible, using trusted vendor images. So let's talk about creating Docker snapshots, and again, formalizing this process. So this is kind of the workflow. So we are all going to do this completely on our on wrap. So the first thing we'll do is we'll start a web-enabled terminal called ttyd. This is a shell that lets us execute things on wrap. And then in that shell, we're going to do a Docker pull. So we'll pull an image from a registry. So we're going to be pulling SAM tools here. Then we'll do a Docker save. And we'll save that to basically our local worker. And that's going to give us a single archive file, so samtools.tar.gz. And then we need to save that snapshot file back into project storage. So we'll use dxupload to get that back into project storage. So again, this is just kind of that in text. But a quick reminder, don't forget to terminate your ttyd instance after you're done doing this. Otherwise, you're going to continue to get charged. You will get an email reminded you at 24 hours, like if you've left your ttyd instance running, but that's the only warning that you're going to get. So please be aware when you use ttyd of terminating it when you're done with it. So what is ttyd? So this is an app that's available in the tool library. So you can access that by going to this tools dropdown here. And here we've searched in the tool library listing for ttyd, and we pulled it up. So when we start our ttyd job, the way we access the shell is at this worker URL. So this will basically open a shell on a worker on wrap, and we can do like our Docker work with that. Just keep in mind that ttyd, there's a little bit of initialization that happens. So even if this worker URL is immediately available, it takes a few minutes for this to start. So if you get an error here, just wait a couple minutes. Like Anastasia would say, go get a cup of coffee, come back, breathe, and then start your ttyd instance. Okay, so the first thing we're going to do is we're going to do that Docker pull. So we're going to be pulling the SAM tools from the SAM tools images. And so that's, you can see we do a Docker pull, and then key.io, and then this is kind of the owner of the image, and this is our image name, which is SAM tools. You'll notice that after the colons here, there's more text. So this is actually a tag. So when you're doing versioning with Docker, this is actually a key thing, is to basically use tags that are tied to specific versions of the software. This way, when you kind of repull your you know exactly kind of what the dependencies are. So we've done that here. So basically, when we do a Docker pull, you'll see that we are actually pulling what are called layers. But when we do that and we use the Docker images command, you'll see that on our machine that we have this container called key.io/containers/samtools with that particular tag. Sorry, I'm just getting used to on24. So once we have that image in TTYD in our instance, we can save that. So this is going to save it to a file in our on our TTYD instance. So we can do a Docker save, and then we need to give it kind of the full kind of path, and then we pass that into gzip, and then we can generate our single monolithic image here. So this is going to be on our worker. So the last thing we need to do is we need to get that back into project storage so we can use it. So we'll do that with dx upload. So we're on a TTYD instance. We've saved our snapshot file to that instance. We need to get it back into project storage. So our main tool for that is dx upload. So this will upload that. It will upload that single archive file into the project. So, okay, now we built our snapshot file. Now, how do we do work with it? So I'm going to show you how to run Docker with Swiss Army Knife, which is an app on the platform. And then Andre will take over and show you how to kind of run Docker as Docker within an app. So this is kind of the workflow for doing batch processing with Swiss Army Knife. So I'm giving you an incomplete recipe here, partly because I want you to experiment with this, and partly because of the structure of files on in the bulk folder in UKB wrap. Well, I'll show you kind of some of the reasons why. So the main idea is we can basically use dx-find data to find all of the files associated with a particular data field. And then we can pass that into basically a for loop that will generate a job for a Docker job for each of those files. So in the interface, so here you can see this is when you're running Swiss Army Knife. You can see that there is an input here for public Docker image identifier. So you can put your link to either your URL or, again, what we're recommending is that snapshot file here. So you want to put a link, basically the location of your Docker image or Docker snapshot file here. So when you actually run it on the command line, so again, we use dx-run to execute with apps. So here you can see that there is an input called image_file, and this is where you would supply that snapshot file here. So just a quick overview, so we'll run Swiss Army Knife, and we're going to pass in this VCF file from our data folder. And we'll pass, we want to run samtools count on that. This should actually generate an output file, but I just wanted to kind of give you the basic idea. So I want to talk a little bit about dx-find data. So this is probably one of the power tools on the platform is basically working with dx-find data. Here I'm showing you that how to pull to basically find all of the files that are associated with a particular field ID. So if you use this -- property and then pass in the field ID with field underscore ID equals, so in this case we're doing 23148. If you give it a -- that's going to return only the file IDs. So this dxFindData command is going to return you all of the PVCF files that are associated with this data field. So one thing, and this is why what I'm showing you is an incomplete recipe, is that you'll notice if you actually pull these the files that are associated with this field, there are PVCF files, and then there are the associated .tbi files with them as well. So when you're doing an analysis with both, you need to have both of these pairs. So I'm not going to show you how to do this right now, but I think this is kind of a good exercise to kind of work with. This might be more actually appropriate to kind of use in an app, which Andre will show you in a little bit. The other thing I will tell you is be very careful with dxFindData and with fields that have 500,000 files. And so these are files for like all of the individual participants. I would say focus on only those fields that are for aggregated files. So the PVCF files, you don't have one PVCF file for each participant. These are aggregated across the entire population. So in general, be very careful with using dxFindData with those singular files. So we have a list of files. So how do we do things with them? So, this is going to be kind of a whirlwind tour of kind of Linux or Unix commands. And one of our best tools for that is a command called xargs. So if we pass in basically a new line delimited list of things into xargs, it's going to let you loop over that. So this is a recipe that I use all the time. So this first part, this is a placeholder. So this is going to say that you're going to loop through each of these. So it's going to go, the first iteration is going to be 1, and the second iteration is 2. So, and then we're going to basically open a subshell. So that part is in green here. And then we're going to use that placeholder here to substitute that value into this echo statement. So if you run this, so we've got basically this is delimited by different new lines. You'll see that this basically gives you this is 1, this is 2, and this is 3. So again, this is we basically to kind of start putting things together. We use dxFindData, and then we use -- brief, and so we're going to pass our list of file names into xargs to do something with it. So this is a very complicated command, but let's try to take it apart. So this first line here, this is that dxFindData command that we had. Then we can pipe that into xargs, and then we can use the xargs to run separate dxrun jobs. And again, we have that placeholder. So that placeholder, and we're going to substitute that our file input with that placeholder right here. And one other thing I will say is that you should tag your jobs when you're doing something like this, when you're running multiple jobs. And the reason for this is that if you make a boo-boo or you make a mistake, you're able to basically terminate all of the jobs with that tag, and I'll show you a recipe for that. Finally, yeah, you can see in the dxrun statement, we specify that -i image file. So that's going to be this is going to be basically that file that contains our snapshot. So like I said, I'm not giving you a complete recipe here. I was up late last night basically trying to kind of come up, basically working with, both the PVCF files and the .tbi files. So I will say that this is kind of a partial solution. So we want to select all of those files that that contain vcf.gz. So these are going to be the VCF files themselves. So one solution to that is to use dxFindData and output JSON and process the JSON using JQ. So this is a filter in JQ that will return only those files that contain vcf.gz. So you'll need to basically create a more sophisticated filter to actually pass in both the VCF or the PVCF and the TBI, the index files that are associated with those VCFs. So I mentioned about dxterminate. So you can use dxterminate to terminate a job ID. But say you have a bunch of jobs that have a particular tag and you want to terminate them. So you can use so there is another command called dxFindJobs. So we can find all of the jobs that have a particular tag. We can pass that list of jobs into dxterminate using xargs and terminate that series of jobs. So this has saved my bacon. So this is why you want to make sure to tag your jobs. So we learned a little bit about using Swiss Army knife with Docker snapshot files. We talked about how the -i image file input is how you kind of basically specify that snapshot file in Swiss Army knife. We learned a little bit about dxFindData and how to pull all of the files for a particular field ID. We learned about the thought of doing batch processing. We want to do basically pipe that into JQ, a combination of JQ and xargs to submit one job per file, and then the idea of tagging jobs so we can terminate them if we make a mistake. So Andre is now going to take over. And
okay, thank you. Let's move to the next section. So in the next part, I would like to talk about how to Dockerize DNAnexus applets. So in the next section, I'm going to explain essential applet components. But at this point, I would like to recommend you, and if you would like to hear more about creating applets and workflows on RAP, you can watch our previous webinar and the recording, which is publicly available. And this webinar was called how to create an app or workflow and bring your own tools on RAP. So with that, I would like to talk about basic applet structure. As a first step, I would like to describe and explain a typical directory structure. So typically when you create a DNAnexus app, you must first create a local directory, which would consist of app source code and resources. So that's the typical structure on the left side of this slide. So what we can see here, this is a typical skeleton, which consists of dx_app.json. We will be talking about it in a second. Then we would need directory resources, and then also directory source, which is the place where we would like to create main logic, which is shell script. So to tell you more about dx_app.json, dx_app.json is a file on DNAnexus, and this means application metadata file. And in this file, we can typically find the following sections, such as input spec. Input spec describes individual inputs for our applet. So in this case, we can see that we would like to run some tools and process CRAM or BAM file. So in this file, in this moment, it's a CRAM file. It's a typical class. This is file. Optional is false. This this means that this file is required and it has some name. And then also we can, or we would need to define output file in the output spec, which is now counts txt. And what is also important component of DX-app.json is run spec section, which is telling the applet during the build which interpreter should be used. Here in this case, we are considering Bash, but also typically it could be, for example, Python. And then we are specifying file, which is wrapping all the main logic to the shell script. And then also, I would like to highlight here a part which is called exec depends. This is typically the part of your DX-app.json where you can specify packages. Typical Ubuntu packages you can pull from publicly available repository. So yeah. And on the right side of the screen, we can now check the content of our main logic shell script and what we can actually need to do in the main script. Typically, we would need to first download data. This is the file we are specifying in the input spec section. Then we would need to describe or how output file should look like. And then we can run the main logic, here highlighted with the orange color. We are running some tools, which we actually at this point installed, or we are managing these dependencies using exec depends section in our DX-app.json. Once we do the computation, once we do the main command with some tools, we can then return results and upload results back as an output of our applet. So this is simply a simple skeleton for a typical DNAnexus applet. Yes, and typically you can create this simple structure with the help of DX app wizard command. So now let's talk about how we can actually use Docker images in our applets. During this webinar, we've already seen 3 ways how we can use Docker on RAP. First option was, let's say, pulling a Docker image from public repository. Also, we saw that we can pull a snapshot, a saved snapshot in tar.gz from the DNAnexus project. Or third option might be use it as image for Swiss Army knife. But here, I would like to show you that eventually you can load and run Docker image from the applet directly from the main script, which could be, for example, done as it follows. So we will need to do DX download and download already existing snapshot from the DNAnexus project. Then we will need to use command docker load, which will load already existing Docker image into memory on our local worker repository. And then, this is very, very important, we can eventually run docker run command with the following options. And we can specify, let's say, options --rm. --rm option means automatically remove the container when it exits, after we are done with the computation. And then what is also recommended when using Docker on RAP, you can use -v or --volume. This is the same option, and this allows you to mount local DNAnexus worker directory, like we can see it here in the green color, and mount files to your container so you can work with them inside the container. Another useful option, another useful feature is -w. In this case, -w home DNAnexus, which will set up working directory, the place where or the directory where you are working when you are inside of your Docker image. And then we can specify image name, and finally your command or command of your interest, command you would like to execute for some tools that would be, let's say, some tools view. All right. So, and when incorporating all of this logic into real Applet, that's subject of the next slides. So how we can use Docker images, and let's do here some comparison between DXApp.json with Dockerized solution and DXApp.json without Dockerized solution. So previously, we explained how the typical applet looks like. In this case, it's DXApp.json, and we are specifying in the input spec just input file. And then we are using exec depends to manage our dependencies. But right now, we would like to edit this code and introduce some changes to the code. And now we can consider DXApp.json using Docker. So in this case, we would need to add one more input to the input spec section. In this case, the name would be Docker image. So we are adding one more. And here we can specify the input. And this might be actually our tar.gz snapshot we've uploaded to a parent project or created in our project. And what does that also mean? We don't need exec depends anymore. So we are now trying to rely on the underlying Docker image we have on the platform. This is new concept of managing dependencies. All right. So actually, after this comparison, let's explain. Let's see how we can incorporate Docker directly in the main shell script, in the main logic, and see what we can do to implement that. The main structure or the first steps would be really similar to what we've seen. So on the first line, we would do the download and download our file. But then new section is we would need to do DX download of the Docker image from platform to the worker. Then we would need to load it with command docker load. And as a final step here, we would run docker run command with some particular settings. We would like to mount directory home DNAnexus to home DNAnexus inside the container. Then we are saying, we are telling the Docker that we are using some tools, then followed by particular some tools command, some tools view, and then we would like to also specify BAM file, which we downloaded on the platform. Hopefully, all the logic will produce output file, which will be then transferred back to the project, and our job is successful. All right, so this was a really quick example how we can implement Docker logic into our DNAnexus applets. I would like to also tell you that we are preparing a code base, which we are going to release, and this will be publicly available, so you can then read the code, and try to edit it for your particular apps, applets, and tools, bioinformatics tools. So now, for those who typically develops or who prefer developing bioinformatics pipelines using WDL and DX compiler, here we have a really quick slide. And in short, there is a possibility to specify Docker image in the runtime section of your WDL definition. And if you are interested in getting more details about it, I would like to recommend watching the following two webinars we are seeing here. It's an introduction to WDL on the UKB RAP and advanced WDL concepts on RAP, including Docker. So that would be best for you to see this recording, to watch this recording. So what have we learned in this section? We talked about how we can load and run the mounted Docker image from the Applet script. Then we looked into how to use Docker in my shell script. And then we also showed some examples how to specify a runtime Docker image for your Viddle workflows. All right, so now in the next part, I would like to present even more and some more useful Docker features. And we are aiming at extending existing Docker image with new software. So let's talk about existing Docker image and how to extend it. We will be explaining the following workflow. We will start with TTYD, as Ted told us more about it. We will be pulling image from Docker Hub. Then we will try to run Docker container on TTYD, and we will be working in interactive mode. With that, we will try to install additional software, which is called EMBOSS, inside the container. So these steps will be done inside the Docker container. And then finally, once we are done and once we apply Docker commit command, we will try to save our new software to either our local image repository, or we can save it to snapshot file and upload it back to project for further usage, let's say, for in your applets. So for Docker image, we will be using EMBOSS, and similarly, we can save it to snapshot file, which is called EMBOSS tar.gz. All right, so let's apply some commands. Again, we are working with TTYD, and what is great for TTYD, Docker is actually part of TTYD cloud-based worker, so you cannot install it, or there's no need to install it. It's already there, so you can directly use it. And you can test if it's working properly by running docker-h, which will show you help and all the commands available for Docker. So then what we can do, we can pull some operating system. Here we will be building our new software based on our base image is Ubuntu 18.04. So that's our base image. And by using Docker images command, we can check that our image has been correctly loaded and it's available for our experiments. All right, so now we are on the worker and our image is ready. Now our base image is ready. We are working with minimal Ubuntu distribution. And what does that mean? Many standard libraries are not installed, so we can enrich our image, let's say with wget, unzip, or many standard Unix or Linux tools. So yes, of course, we can do it. But here we are aiming at a demonstration of Docker run command, which now uses -it, which means interactive mode. And Docker interactive mode typically allows us to execute commands at the time of running the container. And we can get a command prompt. And working with, that's exactly the interactive mode. So we can access bin bash tool directly inside the container. And this is also typically a good idea to use it for debugging. So you can debug your tools by running interactive mode, just like jump into the container and see what is happening there. Or you'll also, for debugging jobs, you can run a dxwatch command, or you can connect to your jobs via dxSSH. So for those who are interested, I would recommend to study documentation of dxwatch and dxSSH. So we are in the, let's consider we are in the interactive mode, and we would like to install some bioinformatics tools. We here decided to show you a tool which is called EMBOSS. EMBOSS stands for European Molecular Biology Open Software Suite. And this integrates a range of command line tools for sequence analysis. And this is something like Swiss Army knife. Many of us, we know the tool and we know how to use the tool Swiss Army knife on the UKB wrap, but this is something like Swiss Army knife, but right now for bioinformatics string manipulation. And it includes, it has more than 100 programs, such as Needleman-Wunsch algorithm or working with alignments. So it could be really useful for string manipulation. So how we can actually install this additional software? Let's consider we are again inside the running container in interactive mode. So we can simply run installation commands. In this case, we can install it from Ubuntu library and repository with apt update, apt upgrade, and then apt install EMBOSS. We would also need gzip. With that, if we are successful, the tool is available now inside our container. I would like to mention here that these EMBOSS tools has also web-based alternative with GUI, so you can always compare functionality of the command line interface versus graphical interface. All right, so let's move to the next part. And once we successfully installed our tool, we can now test inside the interactive mode if it's working properly. So in this case, one subtool of EMBOSS is called seek-ret. So we can try seek-ret-h, which will show us help page, all the help documentation for this command in command line interface. Perfect. So now, once we tested functionality of the installed tool, and let's say also we produced some data, we can now decide to commit changes to this already existing image with the new additional software. So which commands we can use actually? So actually what we will need to do as a first step, we would need to jump off, we would need to exit the interactive session. We can use command which is doing exit, which is exit, and then to get more details about the finished or completed Docker containers, we can use a command docker ps, which will list all the existing processes. So with docker ps -a, we can get individual instances of our container, and in this case, we could find container ID for our particular image. And with that, we can then run docker commit with the specific container ID, and this will commit all the changes, all the edits, and installed additional software to new image, which we can tag with the tag EMBOSS with some particular version. In this case, we named it as 660. Once we done that, we can run again the command docker images to check that we now should have two existing images, like the original one, which is Ubuntu, the base for us, and then newly enriched, newly created EMBOSS package with all the new functionality. All right, so now we showed you many steps how to create Snapshot and how to prepare new tools on a wrap in interactive mode. But typically, what you can also do, you can write something what is called Dockerfile. Basically, this is a recipe to install software dependencies, and you can wrap all these steps, all this logic into Docker text file, which is specifying all the logic. We can see some example on the right side. So we are typically building it with the following steps. Typically, it starts with the base image using from. Here on the right side, it's from biocontainers, but in our previous example, that would be something like from Ubuntu 18. Then this should contain all the instructions and about dependency installation, about entry points, and so on. Like we can see it here on the right side, you can, let's say, install software via Conda, and then set up working directory. So basically this contains all the build, make, compile instructions. And this is great idea for your work and for reproducibility and for portability, but typically it's a lot of work. So we would recommend to use others when possible and maybe follow the ideas we were showing. So what we did actually in this section, so we tried to explain how to develop a custom bioinformatics Docker image. We also looked into what is interactive mode and how you can access it and how you can use it for debugging with combination of DXSSH and DXWatch. And then also we mentioned potential advantages of using Dockerfile. So yeah, this was the last slide. So we can maybe now continue with the questions. And thank you all for your attention. So now we have a couple of minutes for the questions.
Yep. So if you have any questions, please ask them in Q&A. So we will we'll try to highlight some of the questions that have already been asked. Okay. So there's a question from Katie. How do you recommend we authenticate to pull from private registries? So Katie found documents about where you put the credentials and couldn't get it to work. So if you post that up onto community, Katie, we'll follow up. We'll definitely follow up with you and make sure that that we could try to solve your issues with that. Let's see. So another great question from Katie. So what is the difference between an app and an applet? Yes. So applets are what you usually develop. So they're only available to you within a project. So they are both executables, and it just has to do with availability. So applets are the things that you develop on your own. If you want to make it globally available, there is a process to turn an applet into an app. That was a great question. Another question.
Maybe, please, may I add one more information for Katie? Sure. Katie, there is already existing DNAnexus documentation page, which is called Developing Apps and Applets. So you can find what are the main differences between app and applet. Yeah. Yep.
Thanks, Andre. Kathleen has a question. So how do you tag your jobs? So is that in DXRun? So yes, so you'll use the -- tag option, and then you'll put the name of your tag after that within your DXRun statement. Great question. Let's see. So I think those are all of the other questions. Andre, is there anything else you'd like to answer?
No, actually, Ted.
Okay. Okay.
I don't have any more open questions here.
Okay. Well, thanks everyone for sitting in on our session, and I hope you've learned something about kind of using Docker on RAP. It is extremely powerful, but we tried to break it down and kind of make the process a little more understandable. So hopefully this will get you a little farther. Chai is mentioning that there is one more question.
There is one more.
So we'll follow up with that question later. But again, just want to again reiterate that all of the code that we've shown here is going to be made available in the repository. So look out for the posting on community. We'll put the link to the repository there. Anything else, Andre?
No, thank you. Thank you very much, Ted. Thank you very much, Brenton. And thank you all for your attendance and for being with us.